Hi, I'm Denny O'Brien, veterinary neurologist at the University of Missouri College of Veterinary Medicine. What we're going to do today is talk about the ABCs of DNA. To understand about DNA testing and how we use this in veterinary medicine, you have to understand the basics of how DNA works. So, you can think of DNA like a book. The same way a book is composed of letters of the alphabet. DNA has its own set of letters. It's a very short alphabet, though. Just A, T, C, and G for the 4 nucleotides that make up all the DNA. Now, the same way the letters of the alphabet are organized into words to communicate a meaning, the nucleotides of DNA are collected into short words called codons. Now, each of these codons is only 3 letters long, but each one can code for a different amino acid. Now, the same way the words in a book are collected together in sentences to contain a complete idea, the codons collected together code for a specific protein, and that collection of codons that codes for a specific protein would be the gene. Now, in a book, the sentences are somewhat randomly collected together into chapters. When we look at DNA, that random collection of sentences, that random collection of genes, is called a chromosome. Now, in the case of the chromosome, the genes, they're not entirely random. There is some rhyme or reason sometimes for why certain genes are on certain chromosomes, but for the most part, it's really very random. Now, these chapters are collected together to form a complete story, a book. And in the case of DNA, that complete story is the genome. So all these chromosomes together, all these chapters put together, make the story of an individual dog. So the genome is the DNA story of that individual, all the genetic material that goes to make that individual who he or she is. For dog breeds, we can carry that one step further. You often have a series of books, like the Harry Potter series, where you have common characters, common themes, and that would be the equivalent of a breed of dog, where the genomes have some common denominators. But just like the Harry Potter story, there are going to be good guys and bad guys in this series that makes up a breed. Now, when we talk about sequencing a gene, all we're talking about is reading the sentence. And this type of sequencing that you see with these peaks of different colors is what's called Sanger sequencing. And that is kind of the gold standard for sequencing a gene. You get very accurate reads of what exactly is there when you do this kind of Sanger sequencing. Now, one thing that's different about the genome of a dog versus a book is that in any individual dog, there are going to be 2 different versions of the chapter. So when the mother forms an egg, one of her 2 versions of the chapter, the chromosome, goes into that egg. When the father forms a sperm, one of his versions of his story goes into that sperm. And when these 2 combine in a fertilized egg, then that makes the complete genome of a dog. But there are always going to be two versions of each chapter, of each chromosome in there. Now, these 2 different versions of each gene are called alleles. And those alleles, those 2 different versions, one from the mother, one from the father, can be the same. And when they're the same story, then they're homozygous. But they can be different. So let's say this is the story about a child getting the animal ready to go to the show. And to do so, they have to give the animal a bath. So if we have a heterozygous situation where we have 2 different versions of the story, then you have the mother telling the child to do one thing and the father telling the child to do another. So the mother is telling the child to give a dog a bath, the father is telling the child to give a cat a bath. Now, when you have 2 different versions of the story like that, one is going to be dominant and one is going to be recessive. So in this case, every time the child gives the cat a bath, he gets scratched. And so he'd rather bathe the dog. And he can get away with it when there's two versions of the story like this, because he can tell his dad, well, Mom told me to bathe the dog, so I didn't bathe the cat, and that's okay. The dog is going to be the dominant allele in that case. And the cat is going to be the recessive allele, the one that's not done. Now, many genes are polymorphic. That is, they have many different forms. So there can be slight variations in the gene, slight differences in what animal gets bathed. Now, we can use those slight differences to tell the difference between individuals. So if we look at dogs, we can see which versions of the story they have heard from their parents. And we can use that to trace these genes as they flow through a population, through a family. Now, when we talk about a mutation, that refers to something damaging DNA, some gamma ray from outer space, or a carcinogen, or just something bad luck with the way the DNA was functioning causes a change. And when the DNA then goes on to replicate, that change in the sequence gets replicated along with the normal sequence. And so now we have a different story moving forward in that part of the DNA where there was a mutation. Now, people who do genetics, genomics, prefer to refer to these things as variants. Say a mutation, we're implying a negative kind of connotation. Usually it has the connotation that something is wrong. But the genomics folks prefer to refer to all of these as variants, changes in the DNA, changes in the gene, and then make a judgment call later on about whether they're pathogenic or not. Now, that's because that mutation may lead to a new functional allele. Okay, so now we're going to give the cow a bath. Well, the aim of the gene is to get the animal ready for the show, and as long as we get an animal bathed and get him to the show, everything is right with the world. But sometimes that change may interfere with function. We end up with a gene that doesn't work anymore. We don't get the animal bathed and get to the show. And when that mutation, that variant, is pathogenic, it interferes with function, then we can get disease. These are referred to as pathogenic variants. So I hope this gives you a little bit of background, a little refresher course in DNA and how it works, that you'll be able to understand the lectures about DNA testing a little bit better.